This is a little addition to Lecture 6, which I forgot to include before, but I want to include it here because I think it's helpful to know. And the previous part of the lecture was to categorize mutations in terms of the nucleotide changes in the protein product and so on. But there's another way of naming the kinds of mutations, categorizing them, and that's based on the actual function. So whether a protein product is able to do its job, or whether it's even present or not. And so there are two main categories, and then there's subtypes inside those categories. But there are loss of function mutations, so that means there is less of the protein, or it's less effective, or there's no protein and it's not effective at all. And there's gain of function. And the gain of function means there's more of the protein's activity than normal. So for example, it's expressed too much and you have twice as much protein as you should have. It may persist even when it's not needed anymore. So many proteins are needed for short periods of time, say during development or during, your metabolism and so on. And then they get degraded and the amino acids recycled when they're not needed anymore. But there are certain sites on the protein required to be cut or tagged to be degraded. And if those are missing, then the protein keeps on going, doing the job, even when it's not needed anymore. So it's too much of the protein. And then proteins can also be expressed where they're not usually found, or changed, turned on in the digestive system when they're supposed to be active only in the lung, for example, or something like that. Or the protein is supposed to be found in the nucleus, but now it's in the cytoplasm of the cell. So it's in the right tissue, but it's not in the right place in the cell. So, and maybe it's interacting with other proteins and doing something it's not supposed to be doing. Yeah, and so those are the general categories. And so then we'll just take a look at how this connects to the types of mutations we've already talked about. So loss of functions, so this could be something like a regulatory mutation where the promoter has been deleted. And that would be there's no protein product at all. So if your promoter is gone, your RNA polymerase can't bind and you're not going to be making an RNA, so therefore there's not going to be any protein. And that is referred to as an amorph. A means no, and morph means shape or form. So it basically means no protein or no function. The protein could be produced, but then maybe it doesn't do the job at all. And so there's a lot of nonsense mutations that will give you that phenotype. You have this truncated protein. It's only maybe the first half of the protein, and it's just completely incapable of catalyzing that enzymatic reaction or whatever it is it's supposed to do. And even a few missense mutations fall into that category as well. If you have one key amino acid for the active site of an enzyme, the active site is the exact place where it binds to the substrate and actually performs whatever the catalytic reaction is. And if that amino acid is changed, then it's not able to bind the substrate and do its job. So those are examples of amorphs. But there's also hypomorph. Hypo means under, and so this means there's less of the function. So the protein is there and it's doing the job, but it's not doing it very well. And a lot of missense mutations give you that. So you've got an amino acid change somewhere in the protein and it causes it to fold a little bit strangely. And so the active site isn't quite as good, quite as effective at binding its substrate or performing whatever the job is. And so it kind of struggles along, but it's not able to do the job very well. The gain of function has categories too. So there's certain kinds of regulatory mutations that can lead to hypermorph. Hyper means over, so that means too much. So the protein is produced more than normal. The protein persists, as I said before, it can't be cleaved, or it can't be, it can't be cleaved, or it can't be broken down or recycled, or shut off when it's supposed to be shut off. So this could also be related to regulatory mutations, or missense mutations most of the time. Could also be nonsense, depends on the mutation. And then a protein is expressed where it's not usually expressed. Spelling mistake. I never see them until I'm giving the lecture. Or it's located, as I said, in the cell where it's not supposed to be. And there's a number of sort of chromosome rearrangements and those kind of things. We won't talk about them till the end of the semester, but this is where we often see neomorphs. So if the wrong regulatory sequence is attached to your gene, it turns it on in the wrong kinds of cells, or two genes are fused together and part of one gene and part of another gene is producing this protein that moves to the wrong place in the cell and starts activating things it's not supposed to. And so it leads to a phenotype because it's doing something abnormal. Loss of function mutations are usually recessive, and recessive means in the cell both of the copies have to be mutated in order for you to see what the effect is. This is not always the case, and we'll talk later in the semester about some of the exceptions. And gain of functions tend to be dominant. That means that you only need one copy of the mutation to see the physical effect. And we're just going to look at an example of that. So this is just an example with fruit flies, my favorite organism. And we're just suppose that this gene is called cinnabar, and the little plus means it's wild type functional. So you've got two functional normal alleles. The enzyme is produced. I've left out the messenger RNA step here. And so you've got, tons of the enzyme, and you've got the normal red eyes of the flies. Here's a loss of function mutation, so it's either not producing an enzyme or it's not producing a functional enzyme. And so this would be an amorph, but this works the same way with hypermorphs too. But you still have one wild type copy, so you still have at least some of the enzyme. And remember, some of you will have already taken some of your MBB with your biochemistry and molecular biology courses. Enzymes get used over and over again in the reactions that they catalyze. So most of the time, one good copy of a gene is all you need, because it's going to make many copies of that enzyme. It's not just one RNA, but the gene can be transcribed multiple times, lots of RNA out in the cytoplasm, translated to make lots of protein, and then the proteins can be reused over and over again before they finally get degraded. Okay, so you only need one copy of the wild type in order to have a wild type phenotype. But if both of the copies are mutated and neither one is encoding a functional enzyme, now you have your mutant phenotype. And you can see that, and it turns out that in the case of the flies, the eyes are a bright orange color instead of the sort of dull red. So you actually see a physical phenotype when you have both copies mutant. Neomorphs and hypomorphs, the gain of function mutations are usually dominant, and again, there can be exceptions to this. But just imagine you've got a wild type gene A, and there's a normal amount of protein being made by each copy, and so you have the wild type phenotype. But if you have a neomorphic mutation, let's suppose it's a hypermorph, and it makes more protein than normal, having a wild type copy doesn't fix that at all or compensate for it. It actually means that now you've got, instead of sort of 2 units of protein, suppose that was 100 copies and 100 copies, now you've got 300 copies instead of the normal 200. And so that can give rise to a phenotype, right? Because of having too much of the protein. So when you have only one mutant copy, you can actually see the physical effect of that in the organism. And if you have 2 mutant copies, now you have, instead of 200 normal units of the protein. Now you've got 400 units. So it could have the same phenotype, or it could have even a more severe effect. And we often see with this kind of mutation that when you have two bad copies of the gene, you have a more serious phenotype than when you have just one copy. And that's all I really wanted to mention. We will see examples like this later in the semester, especially when we start talking about sort of genes working in pathways, encoding enzymes, and when we talk about chromosome rearrangements and that sort of thing. It'll tie back to this topic. Okay, we will see you in class.